with explaining the phrase whole genome sequencing. As the phrase tells you, it can determine the organism's entire genome by uncovering the order of the DNA bases, especially with the help of computational techniques. Before, the techniques used for whole genome sequencing were shotgun sequencing, Sanger sequencing, and these had resulted in the first human genome um, uh, that was completed as a part of the Human Genome Project in 2003. But now the next generation sequencing techniques like PacBio, Illumina, Oxford Nanopore, etc., have now been able to increase the efficiency, decrease the labor, and as well as decrease the costs. So that's good for everyone. With all of this in hand, we are now hoping that in healthcare and research, these next generation sequencing methods would become even more frequently used to identify genetic differences, variations, diversity, and so forth, would help in much more effective diagnosis and treatment. So yeah, here you see the Oxford MinION device, which we'll talk about later. So in this project, since I was not able to access the facilities of the lab, I did most of the work from home, but it was still very important to understand how I got the sample data. So in my project, I was able to analyze the signal data after the processing from the Oxford Nanopore device of human chromosome 10. So explaining what nanopores are, it's pretty simple in its name, you would think, but it's actually a lot more interesting. So like I said before, it's a next generation approach, but some people say it's actually a more third generation approach for genome sequencing. It's able to sequence or read a single molecule of DNA or RNA without the need of PCR amplification or chemical labeling techniques. And even though I wasn't able to witness this in person, I have seen enough videos and demonstrations in the lab to believe it completely. So yeah, Oxford Nanopore uses biological nanopores. I'll explain just what it means. Biological nanopores here is different from, okay, first off, biological nanopores, as it sounds, are transmembrane proteins, and these are called porins. And what Oxford Nanopore uses is something called alpha hemolysin, which is an extremely stable transmembrane protein found in bacteria. These pores are embedded in electrically resistant lipid membranes. And these lipid membranes actually serve as porous membranes, and thus limiting the size of the molecules that can actually pass through them. So the other type of nanopores, which is nanometer-sized pores, are made in various metals or metallic alloy substances. Now, this is not yet ready for commercial purposes, but yes, it's pretty much in the development. So that's something. This technology that was used here uses the process of electrophoresis to transport an unknown sample of whatever we're using through a very tiny orifice. So this is exactly what happens here. As you can see, So this is exactly how the sequencing process is. The speed is really fast, and the accuracy also is a lot better, even when you don't use anything to improve the efficiency. While passing through these different nanopore channels, as you can see on the bottom right, these are the nanopores. The samples, when passing through these nanopores, would cause characteristic changes in the density of the electric current that is in the nanopore. And this, so when there's a change in density, that part is recorded of each base, and in real time as the DNA molecule passes, providing a readout of the sequence. sequence. Now, this is different for each DNA base because of the different shapes and the molecular interactions. So this enables really fast processing in real time, which is efficient. And after that, we can do data analysis on its Oxford Nanopore platform. So here is actually the DNA sequence that this is after library preparation, when we have added the motor protein and the adapter sequence to the DNA molecule over here. The motor protein, actually, there's a tether in the lipid membrane of the Oxford nanopore, and there the motor protein attaches to the tether, and then it walks on the tether, bringing the adapter sequence close to the nanopore, and then the process continues like we saw before. And here is a little more of a slow version of the entire process. You can see each DNA being read like one after the other. So yeah. So as I talked about before, it is quite simple as compared to the other methods. Very few reagents are required, most of them for library preparation. The power required is really less. It's a long read sequencing technology, and so the assembly is quite easy. And also, because we can do simultaneous processing, it is possible in this technology. Repetitive genome sequences can also be spanned with minimal gaps, and also large structural variation can also be easily found. Error rate can be quite high, especially if the library preparation is not done properly. But even after that, we can do multiple processing of the same sequence, and that would like decrease the mistakes of the software. Okay. So talking about the whole genome sequencing workflow that I followed for the entire project. So first off, we start with the library preparation. Then we have a step called Basecalling, followed by alignment and assembly, variant calling, and finally visualization, annotation, and analysis. So this is how the project would have progressed had this been a normal scenario. But since we were in COVID, my access to the lab was not allowed, and I wasn't able to do any library preparation on my own. And my project probably wouldn't have been possible had I not found this lifesaver software, that was software pipeline, which I will show right here. And this was the Nanopype substitution. So the center portion, which is the processing region, was exactly what I needed to analyze the data that I got after the the nanopore processing from the Oxford devices. So the library preparation, talking about the library preparation, even if I didn't do it, was a requirement, because no matter how clean we think the nucleotide sequences are, it still would need some amount of purification processing before we're able to put it into the device and then finally realize that we have the DNA sequence for analysis. So the steps roughly included splitting the source genomic material into the required lengths, conducting the end repair, attaching the adapter sequence, the motor sequence, ligation of those adapters to attach them, enrichment of the adapter ligated library, and a couple more things to ensure that nothing comes off. So that was the library preparation. After the library preparation, here is where the use of the Oxford device comes in, of which I was talking about so much before. So so I would like to take you back to the first slide. Yes, this is the device that I was talking about. After the library preparation, we pour the library prepared sample into the device, which then would convert the genomic data into electrical signals, as we saw before, as the molecules would pass through the nanopores. So this electronic data would be stored in the form of FASTA files. These are a specific type of files that would then be taken up by the next software for processing. And to not make this overly complicated, I will omit the file types from now on. So as you can see, the MinION is on the left, and that is the smallest component. and it can easily be connected to your laptop to simply get the files that you need from it, the electronic data from the device. So, okay, so here, yeah, we had the storage part, which is how we get the data from the Minion Oxford Nanopore device. Then the processing stage, wherein the base calling and the alignment happens. So there were three different softwares, but I decided to go with Guppy here because it actually was a lot more similar to the software that Oxford Nanopore provided. Now, mind you, I did not have access to any of those amazing softwares that were with the kit because, again, I was not in the lab. So this open source implementation really saved me. For alignment, Minimap 2 was what I ended up using. And for analysis, I decided to go only with structural variation as variant calling, and the software was Sniffles. And finally, I did a visual analysis using a software known as Integrative Genomics Viewer. Yeah, so Basecalling. This is where we simply convert, not we, the software converts the electrical signals to nucleotide molecules, and the electronic data is recognized as being of particular base and is converted into that base. Now, I was really interested in this area of the entire project because it really piqued my curiosity as to how you could even predict what the base you think is going to be, because electronic signals seem such a different thing when compared to the simple ATGC of the DNA basis. So what I found out was initially when a different kind of algorithm was used, the accuracy of base calling was as low as 80 to 85%, which is really bad. And now the accuracy is at least 95%. So this was because earlier hidden Markov models were used, which is an algorithm that basically relied on an assumption that said that if we want to predict the future of the sequence, all that is important is the current observable state. All the other states, other than the current one, have absolutely no impact on what we want to predict. As I think it is intuitively understandable, it would be really good at representing the short range dependencies, but as it turns out, it was really bad at the long range, at capturing the long range dependencies. So that was the major disadvantage of the hidden Markov models and why the accuracy was so low. One thing you can probably think of is that this is quite easy to understand because the current DNA sequence that we have is the one that is used as the prior reference. Oh, reference might be a wrong word. Prior model for it. So you're basing your prediction on your current DNA sequence, and that doesn't, it just doesn't give you the right sequence that you want. So, and this would also result in significant biases because there could be particular characteristics of the DNA normally that maybe that sequence probably does not have. Maybe there are a lot of errors, and that would probably generate biases, and that might not give us a very accurate sequence. So an alternative to this was neural networks, which depended on training data, and so the performance of any neural network would be majorly influenced by the data that we use to train the model. This would help us take into consideration the other modifications in all of the other sequences of that particular DNA, like of all the different ones we had before our sample. And so if there are any base modifications or sequence modifications that are to be taken into consideration, we would have it. So this is how the accuracy was increased drastically from about 80 to 85% to 95% above. So that was the entire base calling. And as you can see, this is one part of the entire, I guess, the file that was called of that particular DNA strand. Okay, coming to alignment, assembly, and variant calling. So I realized that showing a file like this for alignment, variant calling, and assembly did not make as much sense as showing one from the genome viewer. So here we are. I would explain just what is happening in this picture. But first, to understand what alignment is, we would need to understand that it is done to actually have the reads of our sample, that is the different sequences that we've put through the Oxford Nanopore device, like each of the nanopores, each of them is called a read. And so that read needs to be mapped to a reference genome. Comparison of each of the reads to the reference genome and seeing where it fits exactly in it is quite time consuming and would also be computationally intensive. So there's one of two algorithms that an alignment software normally follows. One is called the de novo assembly, which utilizes the overlaps that is present with each of the reads with some of the other reads. This is done computationally, and then these are then taken and then ordered into a framework of a sequence genome. The second one, which is what is used by Minimap2 in Nanopype, which is the software I used, is reference-based assembly, which would build our sequence by mapping each read to the reference genome, such that the sequence that we're constructing is majorly similar to the backbone or the reference sequence that we're using. So here in this image, what we're seeing is the alignment of the different reads that we have to the reference genome at the bottom of the screen right here. So the gray bars represent the single different reads and that have been aligned at a particular place to the genome. So above the reads, you see this graph, which is gray in color, and that is essentially just a bar graph that shows the number of reads that have been aligned at a particular location. And anything interesting that you see here, which is colored, are actually mutations, deletions, insertions, anything else other than the normal read that we see when compared with the reference sequence. And I see blue, red, and I think a couple of greens here and here. Okay. Yeah, the intensity of the color, as we see here, is different. It's not the same for all of the different regions, and that's because the regions that are low in intensity actually show the quality or the confidence of the mutation that the software predicts there being a mutation. So yeah, here we see the impact of a variant calling inside the visualization software. So as you can tell, variant calling is simply just a process wherein the software concludes that there may be some difference between the reference and the target sequence at a particular position in the genome. And it's only done after alignment and assembly of the consensus sequence, which is our sample and the reference genome. So the variants can definitely be of different types. So here, the purple eyes that you see in a straight line are all insertions that happen at a particular position. And the different DNA bases that you see scattered here and there are single nucleotide mutations that happen inside this particular genome. So after all of these steps are complete, comes the annotation part and further analysis. Now, this I was not able to complete during the duration of my internship, but as far as I understand, annotation would have been done in two different ways. Like, it would have two different components. One would be analyzing the structural features, such as the sequence length, the GC composition, etc. Information about the structural of components of the genome. And then the second part would be prediction of the different genes that would be inside the genome, because not all the parts of the genome code for proteins. So this would lead to the identification and location of gene structures, coding regions, and regulatory regions. So I remember the tool for this being different for prokaryotes and eukaryotes. So I was going to use this tool called Augustus, and because I was going for human chromosome 10. So yeah, and after gene prediction, 1 second. So gene prediction, yeah, then we have finding the similarities in the functional aspects of the genome that we did using BLAST. So functional similarities, for example, would be to understand the gene cluster for the secondary structure, for the secondary metabolites that the protein would produce, finding the transmembrane domains in the protein sequence, maybe some pathway information, etc., etc. So this was what I might have done. So, but in conclusion, what I learned from this entire project was that Nanopype can easily be used for small signal analysis, and long read sequencing would offer a much more simplified genome assembly, because it just takes up a lot less time and effort and resources. And we also have the ability to span repetitive repetitive genomic regions with as less gap as possible, because we can simultaneously or repeatedly analyze the genome sequence. Integrated Genome Viewer was a very effective tool for visualizing the genetic variations like mutations, SNPs, indels, all of which we saw. Sequencing molecule per molecule would remove the necessity for having PCR amplification and the errors and the problems that that would cause, and it's subsequently simplifies the library preparation while also giving a much higher read length and throughput. Capital cost is low because there isn't much needed for this. And I think it is quite positive to say that it may be possible to provide personalized, detailed diagnosis and treatments of genetic diseases in the near future, as the understanding of genes just keeps increasing. And we get to know even more on interwoven characteristics of different genes, even when they are at different locations. So thank you so much to my viewers for listening to me so patiently. I would also like to thank my prof, who actually gave me this opportunity and then guided me through all of this. And finally, thank you so much to the Biotech Research Club for giving me this opportunity. It feels great to be here after such an ordeal of social isolation. Yeah, thank you so much. I would be open for questions.